I am Carlos Loucera. I'm going to present towards a metagenomics interpretable model for understanding the transition from adenoma to colorectal cancer. I work at the clinical bioinformatics area for the Fundación Progreso y Salud and the Instituto de Biomedicina de Seville. I work with Carlos Sanchez and Maria Peña, who have also contributed to this work as the head of our lab, Dr. Dupazzo. I'm thankful for the ISBM and the CAMDA conference proceeding, and here we are in Montreal, also called Seville. In July, we are over 30 degrees, near 40. I'm from the north, so bear with me. This is like hell for me. I'm going to start with a brief summary of the talk. First, the problem. We try to classify samples as healthy or colorectal cancer. Okay? We are going to use a machine learning pipeline in order to do so, using functional metagenomic profiles. So we are going to start with a bit of history, then how we understand the interpretability of the model in the context of precision medicine, then the experimental design that we have carried out, the model using explainable boosting machines. Then we are going to talk about the interpretability from the feature selection stability analysis, and once those features are selected, how we do rank them, and are those ranking stable. And then we are going to have a brief summary of the results. So here is a world of clouds, a cloud of words, okay, history bytes. So this is just the corpus using the last papers that we have used, metagenomics, the Slack chats between our group, and as you can see, we talk about machine learning, about CAMDA, about functional profiles, and interpretability. So, in the context of precision medicine, what is interpretability? So first, we have to adhere to biological driven hypothesis, and then we have to check that our model conforms with this, and also we have to validate it with the literature. Then we have the relevance scores that we all use, but we need to say something more about these relevances. This is like putting metrics around the interpretation. Okay. Then we have the sample-wise interpretability. We need feature-wise responsibility. We need to tell how are we predicting a sample, a patient. Okay. We need to describe how the decision was accomplished by the model, but also if the model takes bad decisions, that in the future maybe we can check these bad decisions, see what the model is saying about why he took such a decision, and maybe we can find patterns and have a feedback loop with the training and improve our model. And this must be fully automated from the beginning with the functional profiles, with the raw reads, to the functional profiles, to the machine learning, to check the performance of a model from 2 points of view, from classification metrics, but also from the interpretation part, doing data-driven tests. Then we have the biological interpretation of what the model says. So here in Seville, we work at the healthcare system. This is a great risk environment. So when you say that a patient a sample does not take a treatment, you have to say something more that the machine said it. Okay, you need to tell the patient why. So this is our experimental design. First, we try to address the classical questions. This is all performance metrics, okay, across project analysis, intra-project analysis, and leave one project out design. Then we are going to check the model interpretation. So can we trust our model beyond metrics? So how stable are those selected features? How consistent are our rankings? And if a condition was not seen during the training for the model, can we say something more about this condition? In our case, this is adenoma. Okay, then we have some sort of biological interpretation. I am not a biologist, no a bioinformatician. I am a machine learning researcher here at the lab, so I'm not going to talk much about this because I can only try to repeat what they say to me. So we have a literature validation of the model we have constructed. This slide is very important for me, because this is the slide that we use at the first CAMDA challenge that we participate. So the idea behind this is the idea behind our current analysis. When you have genomic data and you try to perform some functional analysis, traditional methods tries to put human knowledge before the interpretation. Okay, what we say is that from genomic data we go to functional analysis, and the feature selection is done by the model without any input from humans. Okay, so no human bias here. The interpretation part is going to be at the end. So in our terms, or more specifically my terms, is do not break the pipeline. Because if we filter a species or incorporate cross-dataset analysis into our model, we should say that our machine learning model is guided by an oracle. This is easy to check in competitions because you have unseen data, but when you are working with your own data, with public data, and you write a paper, usually you put your biological knowledge and you put also what you learned about all the data sets that you have and put this information into the model. I think that in order to have a fair comparison, we are going to use to prepend an O at the beginning of each model name to refer when we are using an oracle. When not using an oracle is end-to-end learning. So the pipelines that we train, each parameter is learned during the training for all the training steps and splits and validations. So here is the pipeline. It's very easy. So we start from the functional profiles, then we rescale the data using a logarithmic approach, then we perform feature selection using an FDR correction, discretize the data, and then we use the explainable busting machine in order to have a probability score for each one of the samples, and also a relevance metric that it serves to rank the functional profile. And we have also sample by sample relevances. So what is the explainable busting machine? It's a very recent method, but at the end of the day it's a general additive model. So we have a link function for the labels, a bias term, and then we try to learn one function for each one of the variables. So the important part here is that the impact of each feature can be visualized as a graph, and this leads to interpretability. So what does Diagonalism? At the end, it has each shape function is an ensemble of gradient boosting trees. So we iterate through the features in a round-robin fashion. For each one of the features, we train one small tree, one solo tree that only sees this feature. Okay, then we compute the residuals, and with the residuals and a new feature, we train another tree and update the model for each one of the features. So we update the residuals after each trained tree in a boosting-like fashion. You can say, okay, but maybe if you change the order that you are going to cycle around the features, the model is going to learn something different. This is a comparison. This is using such a small learning ratio that the feature order does not matter, and it's a very clear idea. Then we iterate through all the data thousands of times, and then for each feature we summarize all the trees together. So at the end we have a graph for each one of the features, and we can add them together, all the graphs, and we have the final decision. So in our experience, it has several advantages. For example, it has a minimal parameter tuning. This is very important because we are performing a lot of trading, tests, validation, splits for stability analysis, performance scores, rank stability, leave one out projects, a lot of training. We have confidence regions for each learned feature. This is by construction because we are averaging what a set of trees have learned for a specific feature. By construction, the relevance are additive, so they are very easy to clusterize. This is very important because, for example, in KEGG, using the KEGG orthologs as functional profiles, what we have is a hierarchy that we can exploit. And it also has the option to add feature interactions directly into the model. But bad news, interactions are inflexible in high-dimensional data scenarios because it takes too long to train. The training is not cannot be carried out yet on the GPU, so it's slower than using gradient boosting trees as the extreme boosting machine training over GPU. And more consistent attributions imply that more features need to be interpreted. And in our experience, we have that in high-dimensional, low-sample size scenarios, the commonplace here at the biology field, without a feature selection pre-trained, implies that we need to tune the hyperparameters. So a learning feature, this is a graph example of the top of the ethno profile. Okay, so we see that with linear models we have the coefficients and the sign. Well, here we can go further and see that the positive class is being cancelled. Okay, so if you go up towards 1, you are trying, you are more closely of being cancelled, and the opposite is true. And here we see the cut point at 1, and we see that if you have this feature high enough, you are more close to being classified as cancelled. So in the interpretability part, we are going to talk about the stability analysis following the Novelia paper that is very well written and very easy to follow. So we have the problem that we try how robust our feature selection technique is against the stochastic nature of the methods that we use, the noise in the data, and the subsampling scenarios. So at the end, we try to answer, do small changes in data lead to large changes in our feature selection techniques? Okay, so they propose to define a minimal set of mathematical properties for stability. Then they propose a metric, a statistic that complies with all those mathematical properties, and they use an estimator of a population parameters. So you can subsample various time your data set and try to gather a statistic. Then you have for the first time ever a confidence interval over a statistic in stability selection. They also provide the strength of agreement that is very close to effect size in other disciplines. So we go from bad to intermediate good to excellent with two hard thresholds around 0.4 and 0.75. Okay, so here we have the stability analysis for each one of the data sets that we are using. There are 7 data sets, and the blue line refers to the stability analysis when you use the cross-validation schema, that is 20 times 10⁴ cross-validation. The red line refers to the Nogueira's proposed splitting criteria. The vertical bars refer to confidence intervals. You see that they are very low, except for one data set that is way below in the stability score. This is the Hannigan one, and it refers to when you check the literature, this dataset has the lower scores when they try to predict it. This is because it has low quality, low depth reads. So what we are seeing here is that we have a data-driven hypothesis test, okay, that we can use to debug our data. Here we see the same schema for the ethno profile, and we can see that it follows a similar trend, and again the Hannigan is worse than the other datasets, and that they score higher. So now we have our feature selection. Okay, we have several feature selectors. Most biological papers nowadays, those that use machine learning, their interpretation is driven by the rankings obtained by a machine learning method. But how stable are those rankings? Most of the time, you check just one time the 10 or 20 most important things for your model. You check that half of those are in accordance with your biological knowledge, and you say, okay. So what I say is that we need to go further and ask more about our model. So here we propose to use the rank biased overlap that is also used apart from the original field, that is retrieval analysis in web searches. Okay, we can they have been using it in, for example, genomic annotation, etc. So our proposal here is that we perform the same splitting scheme that Nogueira proposed for interpretability, for stability analysis. Then we compute the RBO distance matrix in the same paper as the RBO, the rank biased overlap. They have a mathematical proof that 1 minus the point estimate of the RBO is a distance. Okay, So at the end, we have a distance matrix between all the rankings that we have carried out across all the splits. And here we have the distribution of those rankings across all the datasets. And we are seeing again the Hanningham problem. So maybe we have a dataset debugging system. And we are also scoring the interpretation part from a data-driven model. So we have checked out the literature, and there is a huge intersection between our model rankings and the most recent literature. So here we have 2 papers that they summarize a lot of these things, and when we aggregate information that provided by our model, for example, for the K-octologs, we can, with a single model, we can go and assume or take the mean for each one of the rankings across all the hierarchy, and we see that the models, that the pathways and the modules that are the best in our model are those that are the best across several studies, and that they are important, and they have been validated in experimental designs of metabolomics. So here I am going to talk about the results. We take as a reference method this baseline from Thomas in Nature at all. Okay, so we are mostly interested is they are going to use the same schema as us, and we are mostly interested for the purpose of this talk, to check out the Leave One Out project. Okay, so here is our taxonomic profile scoring. We found that we have more fine-grained features because we are not summarizing them into species as other papers. So we score a bit higher. Then we have this O-LOPO because here, if we take what the model is saying for other datasets, and we extract what features are important. So we filter out this noise beforehand and remove these features, and we learn again our method. This is the Oracle LOPO, and this is a fair comparison because what they have done before is to put biological knowledge beforehand into the models. For example, they take all the species and they preselect those that are more interesting from a good microbiome point of view, and they perform FDR correction, feature selection across all the datasets, and they use those features to have the final score. So this is a fair comparison, and we improve upon it. And here we have the ethno profile scoring, and what is more interesting here is that they found that when they use gene families or pathways, the scores drop. Here we have the ethno profile that has more fine-grained features, thousands and thousands of features, and when we use them, we improve our scoring, and it is equivalent to the taxonomic model. Here we can visualize, if we use, for example, the K-octologs, we can visualize it using bioinformatic tools to see through the eyes of the model, because we have those relevances, they are additive, so we have the more discredited one, the K-octologs, and then we can go up in the hierarchy and add them together, and it makes sense because the model is a glass box model that is also a glass box model that has also additive features. Okay, so we can, with a single model, we can see pathway importance, module importance, function importance, as many as points of view as you wish with a single model. So at the end, we were interested in seeing if there are adenomas, if there are differences between adenoma, healthy, and CRC. The literature says that using machine learning, you cannot discriminate between healthy and adenomas. Well, we need to go further. We have a probability. If this probability is well calibrated, we can see it as a risk. So here what we propose is that, okay, we have a lot of samples with different labels. We extract those labels that are not explicit CRC or healthy samples. We put them into the test set, and then with the CRC and healthy samples, we divide the data into train and validation. So we train the model with the train split, and then we compute the probabilities using the validation and test set. That is what we see here. For the validation part, we have the healthy and the CRC, and this is going to be close to the reality because we can put a threshold on unseen data, unseen from the point of view of the model. And now we ask, okay, we have a risk score. Can we say that the distribution of the risk over healthy samples, over healthy patients, is lower than the risk that we attribute to adenoma samples? The question is that we can make this split, we can validate it through 1,000 times or 100, and if we perform a mean-width test between healthy and adenoma, what we have at the end is a distribution of the p-values of this test. And this is what we have carried out. And there is an interesting part here, an interesting property, that if we use the taxonomic profile formed 100 times, there are several times that the p-value is not significant. But when we use the functional profiles, all the p-values are significant for all the tests. So we think that this is a very good result. Obviously, we are not saying that we can say that this sample is adenoma. What we say is that we can give it a score, a risk that is, from a clinical point of view, is going to say, okay, this is closer to CRC than closer to healthy. So maybe it is important to check out this patient. Okay, it's a risk score. So at the end, we need to add data-driven interpretation to machine learning projects. Using interpretable models, we can retrieve what is already in the literature, but if we have scores about the interpretation part, maybe we can go further because we have more fine-grained features and stability scores that say that this is right. So at the end, we have multiple sources of information, data sets. It sounds very familiar. So maybe the CAMDA has told us how to carry out experiments under these circumstances. So if it all started with CAMDA, what I can only say is that share, participate, communicate, and enjoy. Don't let short-sighted administrations to take the fun out of the science. And our future work is to include clinical interpretation layer and multi-view profile learning to improve the times and maybe to participate in the next year's challenge. So here are the functional profiles, tools, the machine learning software that we have used, the literature, and our team. Thanks to all of you, and maybe we can meet next year in person. Thank you.